So I'm going to take this microphone as we do a little technology switch here. And I want to thank Dr. Goslin for her comprehensive overview and also the time it takes to keep abreast of all this information. I can share with you all that I've been on a plane with Dr. Goslin, and she has a stack of ALS medical journals in front of them, and she turns the pages faster than I could, and she actually has read them all. So it's pretty remarkable. So, so as we work on this technology change, I want to take a few minutes to introduce our next presenter. And at the end, we will have a chance for a panel discussion and the ability to ask some questions of all our presenters. I want to introduce Dr. Joe Beckman. And Dr. Beckman is a longtime faculty member at Oregon State University. He's the principal investigator and Burgess Elizabeth Jameson Chair in Health Span Research at the Linus Pauling Institute. He's a professor in the Department of Biochemistry and Biophysics, director of the Environmental Health Sciences Center, and has a Ph.D. in biochemistry from Duke. He's published in numerous journals around ALS research and is also a regular reviewer for NIH research grants. So thank you, Dr. Beckman, and welcome.
So thank you. Can you all hear me? So basically, I'm a basic scientist and a bit of a nerd. I really thought that the Big Bang Theory was a new reality TV series. And I'm going to tell you a bit about our work on ALS. And I actually got involved in it not because I knew anything about the disease or had any desire to try to cure it, but rather as Kim showed a graph of the first gene that was discovered, SOD1, I was using the protein made by that to treat stroke. And was trying to figure out, wait a minute, how can this protein that's really protective in stroke be causing so much serious damage? And what's the connection in ALS? And trying to understand what's going on there. And then it's been a pretty good ride for 23 years. And then I'm going to show you some amazing results where we do have a drug that looks really promising. And I know in my own lab we've gone through hundreds and synthesized lots of them. And there's a couple of new undergrads in the lab and graduate students who are here, and they're going to be busy trying to figure out where did I hide it in the freezer so we can move on. I also have a bit of a cold, so we'll hang in there. I did want to come back to one thing that seems really promising, and that's vitamin E. Dr. Goslin mentioned this. So it doesn't work if you have the disease, but it does seem to reduce the incidence of ALS and Alzheimer's. And this is work from Asherio, who was mentioned as one of the trials that, by Steve earlier. So taking vitamin E before you get the disease seems to reduce the incidence. It makes a lot of sense. Your brain is about 3 pounds. It's a mixture of fish oil and butter. It's going to go rancid, and vitamin E helps prevent things from going rancid. So I do recommend taking it. And there's a big backlash these days against taking vitamin E. It's really based on some pretty weak data, but you can get a lot of national press by bashing it. So a lot of the work I'm dealing with really depends upon a lot of new people in the lab, and many of them have gone on to other places. And this is a really fun collaboration. So the woman in front is Ann Roberts, and she was a star volleyball player at Oregon State. And her husband came and was a grad student with me. So he's gone on to Melbourne following her. She was an Australian national champion in volleyball. And they were coming back to do some experiments, and we're using a very fancy machine that costs $1 million and full of liquid helium in the back to measure things very precisely. And they told me about a new drug that we then tried in a different paradigm. And I spoke about this last year at this meeting. And I used the picture of a mouse with a birthday that we had our first mouse that made it over a year. I'm going to show you, this is a very severe model of ALS, that actually we have a lot of mice now that made it past a year. When the Ice Bucket Challenge was going on, it was kind of frustrating to say, wait a minute, I actually have a treatment and it's working if you're a mouse. And this is the drug. The image is actually taken from Wikipedia. The secret sauce is a copper in the middle of it. It's held by 2 sulfurs and 2 nitrogens. And it was the last drug in the world that I wanted to work because I've been a big proponent that copper is toxic, and it is. In fact, when we presented this to drug companies, sometimes you get turned down in 15 minutes because they say, you're going to give a heavy metal to the brain, you're crazy. It's basically what I thought. But the beauty of it is it gets into the brain in minutes. It seems to be amazingly non-toxic, and we understand pretty well how it works. We don't know how it's going to work in humans, though, but hopefully we'll figure that out. So the drug is called copper ATSM, and I'll come back to that in a minute of what the copper chaperone is. But it was developed by the group in Australia, in Melbourne. There's actually like 25 people there working on different aspects of it. It works pretty well in Parkinson's disease. And one of the things that's I need to get across is we're really fortunate in the field of ALS that when that discovery for SOD was made, within a year they had taken a gene out from humans and put it into mice. And amazingly enough, the mice get ALS. And they get it within 4 months. And the more you study it, the more remarkable it is of how well it recapitulates the disease. It gets upper motor neuron signs, lower motor neuron signs, ER stress starts early, just a whole series of aspects. But right now, the model is way out of favor. And most people, a lot of people at NIH are saying, oh, we've been wasting our time on it. And the reason is it's such a good model of ALS that no one's been able to treat it. So no matter what you do, you extend the life from 130 days to 150 days to 160 days maybe. In fact, there's $1 million prize if you can extend the life to about 165 days. It's been offered for 4 years and no one's succeeded yet. Okay? So that's the issue, that it's really been impossible to treat effectively. And the people say it doesn't predict the response in human disease, but that's because nothing really works in the animal disease either. Now, I really am a basic scientist, and what I've been studying is the proteins and what they do, and I'm not going to try to bore you with a lot of science. But overall, what the mutations do is they frustrate metal binding to the protein. Mutations also frustrate scientists trying to understand the disease. But what that means is they screw up with the ability of metals to bind to the protein. And the way our drug is working is it's helping it to refold and form a stable protein. So the approach we'll talk about is the opposite of the antisense that Dr. Gosselin just talked about. What the approach we're using actually builds up the protein to quite high levels, higher than anybody's ever seen before, because it's stabilizing the protein and leaving it in its active form. So there's been amazing progress across ALS, and I share Dr. Gosselin's enthusiasm from coming from these meetings. It's remarkable what's going on. We're still at the base of the mountain, but my hope is we're actually beginning to see the top and finding different ways of getting there. And I've been now at this for about 22 years. Our ignorance is just amazing, and the need to move forward is really great. But it is moving, and there's an awful lot of people working on it. So you may recognize this. This was very similar to the slide, or it actually was from the same webpage, I think, that Dr. Gosselin used. So I want to point out that motor neurons are the ones that are in the lower spinal cord that reach out and innervate muscle. And the disease also affects upper motor neurons. But here's the interesting statistic is, out of the trillions of neurons in your brain, you only have 200,000 motor neurons that make contact with the muscles. So there's thousands of muscle fibers, sometimes controlled by a single motor neuron. They're enthusiastic about it. But that's actually not very many neurons. And the problem in ALS is they start to die one after another. And why is that? The upper motor neurons also affect a major part of this. They also are dying, and they're very large neurons. But really, I'm going to worry about this connection of muscle to lower motor neurons more. And part of the reason is that if you think about it, you have a large neuron in your spinal cord, but it puts out an axon or a wire that is over 1 meter in length. So the volume of the cell, it's not just in the cell itself. 99% of the volume of that is in that wire. And then Another huge part of it is in the connections it makes with the neuromuscular junction. So the neuromuscular junctions are actually quite huge and expanded. And those are shown in the bright colors. And in this disease, these things are coming apart and disconnecting and reconnecting. And the muscle itself goes from having this very nice structure of having fast twitch and slow twitch structures to, in the disease, the muscle starts to get very confused. The muscle cells lose their identity. The key point here is that ALS is not a disease of neurodegeneration. It's a disease of failed regeneration. The motor neurons are trying to rewire and take over, and in the process, they themselves die. And understanding that is important for understanding how you begin to attack it and the multiplicity, because any one approach isn't going to work. It's actually coming up with a continuum of treatments. This slide I'm not going to go into, but it's basically to show there's an awful lot of different types of cells present. Dr. Gosselin mentioned astrocytes and microglia. There's oligodendrocytes and Schwann cells, muscle cells, satellite cells. They all release factors that interact with each other, and something in this disease goes wrong where astrocytes that are normally protective start to secrete factors that kill motor neurons. So this is kind of the full employment act for scientists. There's so many different things to study, you could keep busy for a whole career. The trick is trying to figure out which ones are really important and how do you start to sort it out to figure out how are you ever going to treat this awful disease. The other thing that's really amazing in ALS is how fast motor neurons go. So the pink cells that are scattered in the left hand are motor neurons. The holes are the nuclei. And you can actually see in some of these cells they're shrunken and starting to have aggregates. But in humans, when we go to study ALS at autopsy, usually what you see is at the right. There are no motor neurons left in most of the spinal cord. They've died very quickly, they've been absorbed, and then the surrounding cells have formed scars in a gliosis that we also don't understand that process. But those pieces are really important, and why these motor neurons die has been really the focus of the disease. Aggregates are talked about as being very important, and there's some amazing aggregates here, but there's a lot more going on than protein aggregation. And that's one of the battles that we're working on is what does it mean to be an aggregate and how do you stabilize proteins? So for 100 years, we knew about the disease. We had pictures of motor neurons dying, but we had no clue what was really causing the disease. And that was in March. They started a large consortium that worked with a lot of ALS patients, mapped the first gene that caused ALS. And unlike the other 34 that have been identified, where a lot of them are proteins that you've never heard of and you have to look up in databases, this one, there was a huge amount of knowledge known about superoxide dismutase, but none of us had a clue why it would be involved in the disease. So it was a real shocker. How can a protein that's expressed in every cell in the body from birth be involved in this very selective death of motor neurons? Why is it that only 2 to 3% of patients are affected, but the disease looks the same but is sporadic? So is there a connection? There's over 170 mutations. This says 150. I just don't keep updating the slide. So different places where you change something on the protein, and it has the same effect. It causes motor neuron disease. So move one carbon atom around on it, and in some places that's enough to trigger this disease. And yet people can live to be 60, 70, or 80, leading very normal lives before they develop the disease. Okay? So do you see some of the dilemmas that people are facing? Amazingly, you can take this gene that codes this protein and put it in a mouse, it gets the disease. You can put it in a rat, it gets the disease. It turns out that dogs have a form of ALS that's due to a mutation in this disease. And you can do it in zebrafish and fruit flies, okay? So something with this protein goes very wrong that induces the disease. There's other paradoxes. You take the A4V mutation, which causes the most serious mutation or most rapid disease in humans, put it in a mouse, and none of us can get it to be toxic. So it's not every mutation you put in, and we don't know why the most serious mutation in humans doesn't cause disease in mice. But you take another mutation and it turns out to be an outstanding model. So that's basically what I've been spending the last 30 to 40 years of trying to understand why is this the case, and then what's the connection between sporadic and familial. If you don't have mutations, is the connection the same? So working on the protein, it turns out it's a very pretty protein. If you make it in a high enough concentration and purify it, it's green in color. So this is expressing it in bacteria and purifying it. And if we take the A4V mutation and purify it, we discovered that we had a tough time making it in pure form. A lot of it was misfolded, but soluble, but it still made a perfectly good green protein. And we thought about it for a bit, and what we realized was that it wasn't binding metals as well. And so the blue form is the same protein missing zinc, and that turns out to be very toxic. So it's not the mutation that's making the toxicity, it's the metals bound to it. And the mutations are affecting the metal affinity. So we also we can culture motor neurons in a plate. And in fact, these are motor neurons cultured on a feeding layer of astrocytes. And so we've spent 20 years characterizing this and working out the mechanisms to exquisite detail. And if you stain it, these same slides, if you stain and look for fluorescence, the green are motor neurons, but the red underneath are astrocytes. And in this particular case, what we've done is we've made the astrocytes angry, and they actually are inducing the death of motor neurons. So there's a cell in the middle that's blue with a little bit of green. That's a dying motor neuron, okay, being triggered by the angry astrocytes. I like it. This rocks them. Okay, so we've worked out an enormous amount about the death cascades. I'm not going to go into that. What I am excited about, one of our collaborators on this 20 years ago is now the head of the vice president in Biogen going after ALS. And it is huge to have a company that big that's dedicated to going after this disease. It's another reason for hope. We've developed various sensitive markers to start to probe the process. We've worked out a lot of aspects, and now we're digging into tissues and can find, as the disease progresses, that we find more of these markers that are shown in green that we think are driving the disease. And the yellow on the very bottom, far right, that's a neuroinflammatory marker that's very specific. That seems to be, it makes a common trophic factor 10,000 times more toxic to motor neurons. So you can actually start to dig into tissues and find out what's going wrong with these types of markers. The other thing we've used is we've used advanced technology, this mass spectrometry, to start to pull out bits of spinal cord. Sometimes you can use a laser to cut out a couple of motor neurons and then be able to introduce them into the mass spec and actually measure what's going on. So the trick of measuring metals bound to a protein is no one's ever been able to do it from proteins in vivo. Metals move, they're all weakly associated. So we've spent many years building methods where we could take straight from the tissue, inject into a mass spectrometer, and we can weigh these proteins down to a single neutron and tell what metal is bound to it. Okay, so it's a technology trick, but it lets you probe not just is there a mutation, but what metals are bound to it. And I'm going to show you, expanding the scale a bit, that what we're doing is that we're weighing these proteins to a single neutron, so every bump is one more carbon-13 in it. Now, the surprise for us in the animal models is a huge amount of the protein was missing copper. And in hindsight, there was a good reason to suspect it. But what we realized was that we could use a compound, copper ATSM, and get copper into the brain. We had to add one more protein called a human chaperone for SOD, which helps insert copper into the SOD protein. And we had an amazing result. So this shows the copper chaperone. It's a fairly complex protein that associates with the SOD protein, and it has an arm that actually swings over and drops the copper into the right side. So it's a pretty fascinating process to think about the biophysics of it. And in doing that, we found that we could give this drug, copper ATSM, to animals, and we could see that the peak that's on the left-hand side drops almost to zero. And what's happening is the protein, the SOD protein, is stabilizing in its mature form. It's actually accumulating to remarkably high concentrations. It's not degrading, it's not aggregating. And in the pharmaceutical industry, this is known as target engagement. So it's a really remarkable example of doing this, showing that this drug is really getting this directly in the cells that are affected. So what we're looking at here is the survival. So I mentioned the G93A mice, and the blue shows how fast they die. And they die of ALS. Their hind limbs shrivel. They lose 20 to 30% of their body weight in 3 weeks. And it's not that they're not eating, it's just loss of muscle mass. And that's the frustration, is hundreds of drugs have been tried, lots of things have been tried, and nothing really works. Now, I actually gave up on trying to treat the disease. I started to look at what makes it happen faster. So we included this copper chaperone for SOD. We added the extra human gene. So we now have 2 human genes in the mouse. And we started to study it based on work that was done in Texas by Maharaj Sun and Jeffrey Elliott. And here the mice die in 10 days. So you put into a second human gene and suddenly the mice are dying much faster. So that's where we got the idea when my friend Blaine and his wife were visiting, we need to try this copper delivery drug. And it worked remarkably well. So this is a curve that we are hoping to publish, should be coming out in the next month or so. So the untreated are shown in the far left, where they die in a couple weeks. And we have 10 mice that are 570 days old and still alive. They're actually dying, we've lost a number of them, but they're to things like tumors, dermatitis, and it's actually, they're becoming elderly. We're approaching late middle age with them. And I'm going to show a video in just a minute. So it shows mice in a cage. So I hope that doesn't upset anybody, but it's actually a quiz. So I need you to pick out which one does not have the gene that causes ALS, and 3 do, okay? The other remarkable thing is we could take away the drug, and then the mice die at about 130 days. And then if we take them when they have symptoms, we can start the drug again, and they actually recovered quite a bit, and they made it out, some of them to over a year, okay? So this is the first time that anybody's been able to turn off and on the disease in these mice. And this survival basically leaves the neurology community or the neuroscience community, when I've talked about it, they basically have no idea what to make of it because no one's ever seen anything like it. In fact, if you look at weight, and Dr. Gosselin pointed out the importance of weight, the problem we have with this drug is actually obesity. These mice actually get a bit fat. And if we take away the drug shown in the blue line, you can see the rapid weight loss, and then the animals die. And if we start to treat them after the weight loss has occurred, they will recover. And I'm not going to show that curve. So here's the test with the animals. So there's 4 animals, 3 have ALS, the SOD gene. They're 405 days, so well over a year old. And the question is, can you pick out which one has the ALS gene? Which 3 have the ALS gene and which one doesn't? Not quite. So I gave it to my 3 kids to look at, none of which have any interest in science, but they all did correctly identify it. You can tell the difference. So the guy in this upper corner is the one that doesn't have the SOD gene. And part of the clue is their legs spread out a little bit and their toes spread out. They're losing some of the fast fatigable motor neurons. And some of them now, at 560 days, they've started to develop some signs of spasticity, and they occasionally fall over as they waddle around, okay? But this is a pretty cool result. So the advocacy has helped. I actually was startled. It was New Year's Eve when I learned that I finally will have some funding for this work. And we're working through the complexities of the Department of Defense. It's an immense amount of paperwork, but we're going to be going into this huge process of developing the drug, and I have lots of ideas for analogs that might work better. So far, none of them have, but you keep going. But this drug itself actually will be, we're hoping to get in trials. There is a company, the Australians licensed it to. They're going to the FDA, and we're going through this intricate dance of how do you take a new drug to market. And so, you know, we can finally treat mice. I'm very optimistic that this model actually is relevant to humans. One of the big arguments that we have if talking with clinicians is, should you do it in familial SOD patients only, or do you do it in sporadics? Okay? The drug actually works in Parkinson's models as well, so it does more than just work on SOD. It protects mitochondria. Wanted to point out that there are many dog breeds that have SOD mutations and they get ALS, so we're going to work on treating the dogs. I also wanted to warn you, someone mentioned a Popular Science article when we published some initial articles and they show copper turnings. Yeah, don't take copper itself. You can take small amounts, but if you try doing it, it's actually got a lot of toxicity. This compound works very well at getting it into cells with damaged mitochondria, and that's the only place it delivers it, okay? And then finally, there's a lot of steps to go forward, but among the many problems, actually the FDA is an amazing organization to work with. They really do get it, that this is a horrible disease, and they will work with you. But one of the problems, for example, is we make the drug, we made 500 grams of it in our lab to treat. That's a pretty remarkable amount to make a pound of a material in a laboratory. If this drug works, we have to figure out how to make about a car worth of it, a ton. So one of the key things when you're developing a drug is the FDA wants to know, can you make the drug consistently? If they approve it, can you meet the need? When you talk about expanded use, can you make enough of the drug? So if you make your favorite dinner and for 3 people, think about expanding it for Thanksgiving dinner for 30, but then think of what it would be if you try to feed an entire city of 50,000 patients. So the drug synthesis and scale-up becomes a problem. How do you formulate the drug? So this drug, we call it brick dust because it looks like red brick dust and it's about as soluble. So how do you actually get it to go into the body? It's orally active, but if you make it deliver too fast, that could be bad because it spikes and clears too rapidly, but it may cause GI problems if it gets into your intestine. Then we have to work out what dose do you give? So I know in the mice how to do it, but I don't know in humans, okay? It's not a regular drug. Most pharmaceutical industry experts look at this and they go, no way in hell it's going to be a toxic compound. It's a copper compound. And it doesn't bind to a specific target in the way a usual drug does. So there's a mindset you have to get around. We have to figure out how you can monitor and see, did you get an effective dose? And then you have to go through the different phases of the study and figure out how do you get this into trials and what's the best way to test it. But they're great problems to have, okay? And I think that's the final part. You know, there's a lot that can go wrong along the way, but I think we can answer a lot of these things, and so I'm very hopeful of where we'll be. So thank you.
If I could ask our panelists. And got a microphone there. So we want to take a few minutes, and we realize the afternoon's growing long, but we have just an immense wealth of knowledge about ALS and ALS research here at our panel today. And so we want to give you a chance to ask questions, and certainly raise your hand and I can call on you. Yes, sir. So the question is once a motor neuron is dead, is it correct that it cannot be rejuvenated?
Yes, that's correct. Once a motor neuron dies, it can't be regenerated. There is a problem with stem cells, and that exactly what you're talking about, that as a cure, stem cells are pretty far away. To get a stem cell that becomes a proper motor neuron and then reconnects to the muscle is very, very difficult. And then there's the issue of, the environment is still toxic. There's still something wrong in people who have ALS. So there's a long way to go to get stem cells to be more effective.
I think the question was about protein aggregation and cleansing and recycling of proteins. Is that a focus of research?
That is a very big focus of research, but I think, as Dr. Beckman said, it's not the only piece. So I think there is a belief that. One theory is that ALS is sort of a type of prion disease that. Like mad cow disease, that abnormal proteins make other proteins abnormal, and that gets spread throughout the nervous system. So there is a big focus right now on strategies to help abnormal proteins be degraded more rapidly. That's a very big area of research right now, and probably very important. Not the whole picture, but one of the aspects.
One question I have for the entire panel is, there's been a stronger focus on the development of biomarkers and looking at in terms of earlier diagnosis as well as a benchmark in terms of determining treatment efficacy. Is there any real progress being made in that area?
There is a lot of research being done, and I think there's it feels like can you hear me? There's a lot of research being done. It seems like the papers I've seen about that have talked about panels of markers that may be indicative of ALS. I haven't seen anything that's a clear correlation or diagnostic of ALS. You may know more.
I think there's been also a lot of discussion with the FDA on biomarkers, which is even as important for drug discovery. And I know that there's been a lot of things published and still being discussed. One of the trials that Kim mentioned, NP001, had some biomarker work. So there's been a lot of discussion in a lot of the trials, but I don't know exactly where we are on that, but it's all been moving forward and discussed with the FDA, because that clearly is a way for us to speed up trials quicker.
So the cautionary note is that a lot of people are pushing for biomarkers as an easy answer to say whether the drug's working or not, than following things like survival or muscle strength loss or respiratory problems. And what terrifies me about biomarkers is that they will kill projects before you really have gone through, because the connection between the biomarker and the disease isn't very well established. So we have to be careful about checking the validity of the biomarkers, and I think there's still the brute force of just let's see how well it works is going to be still very important.
Yeah, and I think we're still quite far away from having a biomarker that would be a reliable marker that could be used, like PSA for prostate cancer. We don't have anything like that.
Questions? Yes, sir.
Exclusionary, like if you have, is there a grouping? Like if you have 5 of those genes, are you more likely to have ALS? Or are some of them, like if you have one of the gene mutations, you're not going to have one of the other? Do you understand the
Yeah,
question?
so the slide with the 35 genes, those are genes that if you have it, you usually get ALS. They're causative for ALS. That said, a lot of those genes haven't been looked at in detail, so there's not clear data about the whether these genes always cause ALS. But those genes that were listed are usually causative of ALS. If you have it, you usually get ALS.
So if I have a relative, say, that has had 2 of those genes identified, and that's unusual, according to his doctor, he's more likely to have ALS or to
Can you
develop
repeat
it.
your question with the microphone?
If you have
Oh,
a relative
sure.
with 2 of the diseases, 2 of the markers, are you more likely to get the disease?
So those are I don't think that's been looked at. They're all individual genes that have been identified as a cause of ALS.
Okay.
And so I would imagine, based on what I was talking about, sort of genetic load, that if you had 2 genes that are usually causative of ALS, the likelihood would be very high that that person would get ALS.
Thank you.
A question in the back there. Please wait for the microphone.
Steve brought up a point that these things can skip generations, and that would be related to the complexity of the genetic makeup. What I mentioned earlier, that even if you have a gene that's likely to cause ALS, there may be other aspects of your genetic makeup that make it less likely to manifest. So it is fairly complex.
I happened to read an article in just a short little thing in a magazine about a study in Australia at the Queensland University that they did for Alzheimer's, and it used ultrasound, repeated ultrasounds in mice with Alzheimer's, and it seemed to activate microglial cells that would destroy those amyloid beta proteins that are in their brains for Alzheimer's. And it seemed to put them back to perfect functioning. And so then there was just one more sentence about how they were looking into that for motor neuron disease. And have you heard anything about that, or any thoughts on that?
I have not seen that study. I have seen studies for ALS using transcranial stimulation, which is not ultrasound. It's a different way of activating the brain, where that's being looked at for different diseases. I haven't seen anything with ultrasound. I can't figure out how that would have any effect on motor neurons.
So one of the challenges in Alzheimer's disease is they made a mouse model where they overexpress A beta and get tangles and plaques. But it turns out they have to do 3 things at once for the mice to actually show pathology. And so lots and lots of drugs work in the Alzheimer's mice, unlike the ALS mice where nothing works. And so that's been one of the challenges, is the A beta is probably not the story in Alzheimer's brain, contrary to everything you read. And the idea of using ultrasound to activate microglia being activated can do lots of bad things too. So it's, laboratory research. It goes a lot of different ways.
Another question? I think we have one back there, then we'll come to the front. Okay. Here we go. Right
I read an article about vitamin B12 shots. Do those help with the limbs if you do it daily, or I know it's got to be a compounded one, but help for getting maybe some of the aches out of your body?
There was a recent small article about using phenomenally high doses of cyanocobalamin B12 that the data didn't seem very good to me, but and it doesn't make a lot of sense. I mean, the doses they were using were like 10,000 units a day or something crazy, and yeah, and to me, B12 is a really important vitamin, but the required daily doses is something like 12 micrograms. And so another thing about B12 is that the only time that you need injections is if you don't absorb it well. And so most people absorb it well. So I advocate use of B12, but not at those high doses, because it just gets washed out of your system. I can't imagine what a high dose like that would accomplish. The article, the data was not, I don't remember the details of it, but I remember thinking that the data wasn't very good.
Questions up here in the front?
I love the idea you're getting money from the Department of Defense, but I did hear from someone else that actually the reason it seemed like more folks, the military, got it was because of the very fine record keeping of the veterans hospitals, and actually it wasn't a greater number of servicemen.
Well, actually, there's been 3 studies that have shown that. And the first study showed there was a high incidence in one of the campaigns in the first Gulf War. And then there were 2 other studies, Harvard and MDA, Harvard NIH, showed that it is actually before that. Dr. Weisskopf, who did one of the first studies, is doing another follow-up study on that. I have not heard that at all, but I mean, it was significant enough to have the VA for the first time do a service connection for the entire duration of military, because they found that there was a higher incidence even before World War II.
This is for Joe. As far as this article that was in Popular Science in November issue of 2014, I'm just going to read a section here and see what you have to say. It says, while looking at stem cells with the mutation ZANG, found an imbalance that caused the neurofilaments to tangle, which is the cause of ALS. A paper he published in April demonstrates that the damage is reversible once the mutation is corrected. Is that a major problem?
Sure. So I mentioned that, how long motor neurons are. If you think about it, there's a tube, a lipid, the cell membrane. It's 1 meter in length. I'm going to try to stay away from Kim and not infect her. But take oil and bend as you bend your arm. If it's a tube of oil, it's going to break up into little droplets. And you have steel cables that run the length. Those are neurofilaments. And so those aggregates, like I showed a picture of, those are phosphorylated neurofilament proteins. And there's something called spheroids that happens in human ALS, where there's these swellings that are unbelievably huge that are full of neurofilaments. The amazing thing is, and they happen with acute aluminum toxicity in the CNS, you can reverse those and the cells, motor neurons, recover. So there's a lot of different aspects. Neurofilaments are a big player. Biomarkers, phosphorylated heavy neurofilaments and other parts of neurofilaments in blood and the CSF are pretty good biomarkers, are growing. So they're there. They're not unique for ALS is the problem. So it's definitely a big piece of the neurofilaments. I have my own theory I published 15 years ago of tyrosine nitration of neurofilaments, which I'm still very fond of. Not many other people are, but I'll win them over eventually.
Is it an overstatement that it would,
it's probably an overstatement, but there's a lot of reason to hope. And certainly it's a big player in it, and there's a lot of different aspects. It's also known that changes in the neurofilaments is one of the most protective things in the ALS mice. That was published 20 years ago. But it depended very much on the model, and it was a genetic manipulation. It wasn't a drug. So they're big players.
You could have high vitamin E, helps lower risk factors. What's the dosage then is considered high vitamin E?
So there's not a clear dosage, and there's a lot of debate about what's appropriate for vitamin E levels. I think Joe may have a better answer. I think my general recommendation is always not high, because that can cause toxicity. So I would say 400 to 1,000 units a day would be my recommendation.
So the other part of it is 400 units is probably what I would go for. I don't think there really is toxicity. There's a problem with blood thinning for some people. The trick is take it with a meal. Most people take their vitamins with coffee in the morning, and it turns out you don't absorb much vitamin E. So 95% of the US population doesn't get the daily recommended amounts of vitamin E. You don't have to take it every day. It's slowly taken up, and that's why it takes years to build up, and it's also slowly lost. But, you know, enjoy it with dinner. Along, probably with the fish oils will help too.
How collaborative throughout the U.S. is research? Let's say, for instance, Oregon State has their research, and they might be a silo. And let's say Johns Hopkins has a research, and he's a silo, and you feel your research might be in a better direction than maybe Johns Hopkins. And if money is allocated throughout the U.S., could it be a better way to use our funding for research if you think you're on a better path than someone else? What's the collaboration?
So there's both a collaboration and a competition, and a lot of different thoughts that go into it. So I'd like to broaden it. It's not just the U.S., but I mentioned Melbourne, and I took out slides about the importance of my collaborations with South Americans and Europeans. So on a broad front, there's a huge amount of collaboration, a sharing of resources, and going back and forth. But, you know, we're like people in the Big Bang Theory. We have our own theories of what's going on, and, yeah, we'll fight tooth and nail about it for years. And the question is, you know, who will turn out to be right? It's when you can figure out how the pieces fall, or, you know, how do you actually make the things better? And in truth, what will happen is that the right answer was something that none of us thought about. So what's really important is to keep a lot of new people coming into this field, keep exploring different avenues. You know, we showed 34 different genes, and that doesn't begin to touch all the things those genes affect. You know, we've tried a couple hundred drugs, but it's thousands that drug companies screen for different diseases. So, you know, the competition is actually really good. I have one of my favorite quotes was one of my friends from South America was at a secret or a private meeting at Johns Hopkins, and he emailed me back in his Spanglish. He says, "You will love this." So one person gave a presentation, and he says, at the end of it, well, I don't want to be a heretic, but maybe Joe Beckman wasn't wrong. I want that on my tombstone.
I just wanted to give a non-scientific perspective of that question. When I first started 17 years ago, I not only described our very bright researchers as working in silos, but I almost felt that they were armed silos because they didn't really want to work with each other, because I think a lot of them felt they were going to find that treatment and they were going to get the Nobel Prize and go forward. Over time, though, things have happened. I think that they've been forced to work with each other. Some things have happened, like the ALS registry. All those great researchers that we showed were funded, they cannot be funded unless they share their data with everybody. And then something happened last year that has really helped the ALS Association work to force collaborations, and that's the Ice Bucket Challenge. Out of that first $21 million, there's some incredible partnerships and leveraging going on that has not happened before. So I agree that it really has changed. It really is working completely different, and it's very, very hopeful that people are working collaboratively now.
I just go away a little bit from academics. I'm just wondering that what are the advances being made in communication? Because all of us are related with the ALS one way or another. One biggest problem is mobility, which the patients lose. And the second one, which really comes and becomes not only challenging, sometimes even beyond that, is the communication. When the patients want to say something, cannot say it, understands it, wants to respond, but cannot respond. And once you lose your speaking powers, then one of my family members is having ALS, used a computer with his eyes, which I see many people do. And once, now in the last about a year, he's not able to do that. Now he used that alphabetical thing by saying yes or no to the letters. But apparently I have heard that some more advances are coming where computers can be used by your electrodes to the brain. So I would have a question on that. Are real advances being made which are available to the public at the moment or the near future? Because that's the biggest hurdle in the later stages of ALS. Thank you.
So communication obviously is one of the most important aspects of treating ALS. It's so fundamental to existence. So you mentioned some of the equipment that's out there. There's computer-based eye gaze systems, and most patients don't lose their eye movements until very, very advanced stages. So most people are able to use that equipment until advanced stages, though my patients describe it as being exhausting and time-consuming. The area that is moving slowly but is moving is brain-computer interfaces. And I know OHSU is doing some work with that and has some pilot programs with some of our ALS patients. The problem with that is that, a small percentage or a percentage of patients with ALS get cognitive issues, and that makes the brain-computer interfaces more difficult to use. But it's described as being very, very exhausting and requiring a lot of concentration. That's still somewhat in the infancy in terms of developing a reliable pattern of communication with that. But it's moving.
Well, I think that one of the new collaborations that's happening is around assistive technology for the ALS Association. And as Lance mentioned, there's a project that is being funded by the Department of Defense in the DARPA program. There's also some programs that actually last month our association, so we have our president, Barb Newhouse, but we also have two other great leaders, Dr. Lucy Bruin, who's our chief scientist, and Kim McGinnis, who is our chief of care services. And they put together a wonderful workshop in Virginia, bringing the best of any sort of technology that might be used for that. And One was a DARPA project. One was that's being focused at the University of Pittsburgh. One is a project that is using robotic arm that is out of Orange County, California. So a lot has been done. There also has been some great advancements from a person with ALS, Steve Gleason, who has really worked closely with Microsoft. And so the technology, I think, will really be advanced the next few years on trying to get that out to patients to be able to use, as well as continue to work with the government. Steve Gleason was able to get his senator to actually reverse some of the Medicare changes in law for the government to cover some of these devices, and it passed the Senate, and we're hoping it'll pass the House too soon.
Microsoft. I know that they're looking for candidates, and I'm going to be possibly one of the candidates that use it.
Great.
Because they said, well, she doesn't live in the Seattle area. And I said, I'll drive clear across the country. So I know that Microsoft is working on one right now that's about to come out.
Great. Good news.
Yes, Mary. Please wait for the microphone, please.
I want to share with you something going on at Oregon State University at the Robotic Engineering Lab. We're working with some very young students and Dr. William Smart. Yes, his name is Dr. Smart. Don't you love that? They are driving 2 Permobiles from our loan closet with Google Glasses. Okay? So being able to operate a full wheelchair just by eye movement. So I'm going to put a little plug. Hopefully we'll have more information soon up on the website regarding that through Facebook and things like that. But research is happening at OSU too.
Okay, so you had said in your research that copper helps damage mitochondria, and is that the cells that are alerting your muscle neurons to move your muscles? Or what cells is it helping, and why do you believe the copper helps? And pretend I'm Penny on the Big Bang.
Okay. I'm not sure what to do with the last comment because I'll be like Raj and not able to talk at all, I suspect. Mitochondria are basically, they're considered the engines. They produce the energy to drive cells. And motor neurons are absolutely huge cells, and they require an immense amount of energy. And the way they get it is they burn oxygen. And to burn oxygen, they use it at one protein that's called cytochrome C oxidase. That enzyme needs 3 atoms of copper in its active site to work. And the reason our mice die so early is they don't have those 3 atoms. Okay? So the copper ATSM does a really good job of delivering copper into the brain. We don't quite know how the copper moves and how it gets to the cytochrome C oxidase, but we're working on that. But what it does do is the mitochondria do seem to work better, and it helps recover mitochondrial function. And mitochondria are well known to be damaged in this disease as well as in many other diseases. So it overcomes this limitation of how do you get copper into the brain and how do you get it to the cytochrome C oxidase. So does that answer your question?
Yes.
So one final question here, and just some closing remarks. Dr. Goslin, you mentioned sort of a list of many clinical trials, and Dr. Beckman, your potential therapy looks very promising. Going into the future, you've also talked about sort of there's many sort of variants of ALS. Going into the future, we've all sort of heard things about like AIDS and cocktails. Is the future of ALS going to be sort of very sort of different people taking different therapies and combination of therapies? Is that a vision for the future?
so I think that's one of the directions it's going in. And I think what you see with some of the trials that I talked about is using criteria to select out which therapies may be most effective for different patients. And I think one of the very clear ideas about ALS is that it's many, many different diseases and many different factors affecting it. So yes, I think the goal will be to figure out more about what makes the patients different. The other movement is to do drug cocktails. And so, you know, that slide I showed you that had all the different factors involved in motor neuron death, one of the pushes is to combine different therapies that affect the different factors. And trials are being designed that will do that with the hope of speeding clinical trials up and treatments up.
Thank you. So we're at our closing time. I know the panelists will be here for a little while, and if you want to approach anyone to ask them an individual question, but we want to take a moment to thank them for sharing their knowledge and their expertise, and thank them for their hard work both treating patients and moving the research needle forward in the lab bench as well as on Capitol Hill. So thank you all for your time, and we love all of you guys, and keep working. We need you. Thanks, everyone, and thank you for being here. So we have videotaped today's presentations, and soon, as soon as we have those, we'll make them available online.